Okay. Well, thank you very much to Pavel and Jochen and David and all the organisers for inviting me to give this keynote. It's a great honour indeed, and thank you all very much for coming. So my research interests in the last few years, as Jochen says, has been focused on One Health aspects of AMR transmission, and my favourite pet has been during this time the Enterobacteriaceae, Klebsiella, and a little bit of E. coli. Before that, I've worked more on hospital infections and transmission within hospitals, and that work was more focused on the gram-positives, in particular MRSA. But I've moved more into the environmental One Health space, and I'm going to sort of split this talk into two halves. So the first half, because I've got a bit of time, I'm going to indulge myself and just talk a little bit some of the questions which I think are outstanding in this field. And some of the challenges and some of the opportunities as well. I'm going to be focusing a little bit on the plasmids as well, because that's the issue that's keeping me awake at night at the moment. It's a very exciting time to start thinking about plasmids. In the second half of the talk, I'm going to be talking more specifically about our own research in Bath and some of the studies we've been involved in lately, and just finish up with some the very latest unpublished results on plasmids. So you're all familiar with the concept of One Health. Just worth bearing in mind that One Health is a much broader concept than just AMR, and it's a much broader concept than just thinking about how we can increase, protect the health of humans. It's very much a concept whereby we're thinking about the health of animals and also ecosystem health as well. And it's evolved itself from the one medicine approach to incorporate the environment and the animal health as well. So with specifically with AMR, I'm sure this is preaching to the converted, but there's this sense that if we're focusing our mitigation strategies on antimicrobial resistance to what's happening within our hospitals, or even what's happening within human communities, then when we're using all these antibiotics in agriculture, when we're antibiotics and antibiotic resistance bacteria are being conveyed into the environment through various sources, and if we don't take this into account, then we're very much akin to trying to mop the floor up or leaving the tap open. So we need to consider all these sources of resistance and all the resources of the actual antibiotics themselves as well. So just recently, just a month or so ago, the UN, the FAO, the WHO, and the World Organization for Animal Health all got together to produce this summary document, which identified these 5 pillars for research guidance in the AMR space. These 5 pillars: surveillance, interventions, transmission, economics and policy, and behavioral insights, all of which are incredibly important. So my particular interest in what I'm focusing on today is the transmission aspects of AMR. And I'm paraphrasing some of their key bullet points with respect to transmission, but they identify the importance of developing accessible methodologies, so democratizing the technology to identify both the sources and the drivers of AMR between these different sectors. We have to think very carefully about the impact of any particular intervention strategies, infection prevention and control practices on mitigating AMR, and how we can figure out how to prioritize and develop more informed mitigation measures. And there's a big question around how our understanding of AMR transmission, and consequently how the intervention measures will work, will vary according to the setting. So what's true for Southern Europe may not be true for Africa or South America or low resource settings. And it also varies, of course, according to which particular bug and which particular antibiotics you're talking about. So I'll start off by just mentioning that, in a sense, the one prime exemplar of antimicrobial resistance in this One Health setting, and really the example that's mentioned more than any other as the kind of case that we should be really looking out for, and a really a cautionary tale. So this is the case of the spread of colistin resistance. So colistin is a nasty antibiotic. It's more or less phased out because it's quite nephrotoxic. It damages your kidneys, but it's been brought back again because there's essentially resistance to all the beta lactams and so on. So it's not particularly. It is a very much a last resort antibiotic, and the resistance is plasmid mediated. The first colistin resistance gene, MCR1, was identified in under 10 years ago in E. coli in food animals. And then subsequently, very shortly after, it was found in raw meat, not surprisingly. So this is very much a food route into the human populations, and then in patients in hospital. So the biology is kind of interesting and kind of telling in that the gene itself is mobilized by these small genetic elements called transposons, which enable the gene to hop between different plasmids, and then the plasmids themselves can hop between different strains. So we have all these different layers. When we're thinking about transmission, this is a key concept which I'll come back to. So we have all these different layers of it, kind of independent mobility in a sense. And once this MCR1 gene had found its way into lots of different plasmids, then it became stabilized through the loss of these transposable elements. So it lost its mobility. So it became part, essentially, very much part of the plasmid. And it's now very widespread. It's been detected in at least 47 countries in 6 continents. So it's this sort of example which has led to this kind of concept that's reflected in the remits of the funding bodies. This is a bit of text from the JPI IMR 9th call from 2019, and I've just highlighted this one phrase here. So this is in their guidance notes. AMR does not recognize geographic or human animal borders. And this probably very much comes, this view very much is informed by this example from colistin. So we can visualize this kind of free flow across these different settings through the sort of classic One Health figure, which we're probably all very familiar with, and we've all probably used. I've certainly used these sort of figures, and it only took a 5-minute Google to actually come up with absolutely hundreds of examples of more or less the same sort of diagram with all these different settings, with lots of arrows in between them, showing this, giving us this sense of free flowing of AMR transmission from one setting to another. So I'd just like to reflect very, very quickly on how we all have a kind of a model in our heads of how we think the universe works. And when we're used to seeing figures like that, then somehow at some level this will inform this understanding that there is this very much a free flow of resistance in this One Health sphere. The problem with figures like this is they show the flow, they show the arrows, but they don't show the absence of arrows, right? So if there is any constraints to flow, then these won't be evident in these sort of figures. And we have to always sort of be on our guard a little bit of how we allow ourselves to be influenced by figures like this. So I just want to sort of bring forward this question a little bit more explicitly, and just question that statement from the JPIMR guidance notes. Is that really true? Is the flow of AMR across One Health landscapes really unconstrained? And if not, then why isn't it? What are the barriers? Is it transmission opportunity, or is it something that more about ecology and local adaptation? So some strains are more suited, more adapted to particular settings than others, and that imposes barriers on the flow of strains from one setting to another. There's an even more basic question is, well, okay, so we've got these arrows from one setting to another, but what do they actually depict the flow of? What is it that we're supposed to be tracking? So I've already mentioned that these different levels of mobility, so you have the strains, you have the plasmids, you have the transposons, you have the genes themselves. So what is the unit of transmission that we should be most interested in? And given those difficult questions, how then do we take our observations, our surveys, our data on the distribution of the different strains, the different plasmids or the different genes, whatever we're looking at, and from that information, how do we gauge the risk going from these genes, these strains are present in this farm or in this river? So how then do we understand what risk that poses to the human population, and if it does pose a risk, what were the best intervention measures to mitigate that risk? So just to introduce a sort of basic ecological concept, you've probably all heard of the Buss-Beckling hypothesis, which refers to the distribution of things, biological entities over space. And this states that everything is everywhere, but the environment selects. And this is very much the case for bacterial populations, in that there is plenty of opportunities for transmission. But once a strain finds itself in a novel exotic niche, then it has to cope with competing with the native flora that are already adapted to that niche. And we find this examples of local adaptation in bacterial populations everywhere. So there's all sorts of adaptation going on to microhabitats. Many times we don't understand the basis of that adaptation, but there's lots of papers out there that would suggest that if we're thinking about a selective landscape, then rather than have a nice smooth thing where bugs are free to move all over the place, the selective landscape is actually much more rugged, like the bottom picture here, and there are peaks and troughs of fitness, and it's actually difficult to traverse that sort of landscape. A particular case of local adaptation is, of course, the host specialism. And we're all now familiar with the concept of spillover, where bugs can actually move from one host to another. This is actually kind of a difficult thing for many bacteria and other pathogens to do, because of these aspects of local adaptation. And the success of a spillover event depends on all sorts of complex ecological parameters, such as how well the species interact, how similar the different species are, different environmental factors, source host factors, recipient host factors, and so on. So there's a lot of really basic ecology which is really, really relevant, and a whole body of literature which is really, really relevant to this One Health question. So if we're thinking about risk, we can think about the potential drivers of AMR transmission. So we think about the opportunity, how things actually move around and have the chance to transmit. As I mentioned, the ecology, we also have to think about antibiotic usage, of course, and how that might correlate with the level of resistance. Is it the case that if we stopped using antibiotics in farming tomorrow, then all the resistance would drop to zero? I don't think so. We have to think about setting specific drivers, how we treat our animals, and wastewater treatment processes, for example. And then we have different levels of risk, which I've given this kind of traffic light system here of green, amber, and red. So if we think about what might happen actually in the One Health setting, the different types of transmission there are. So you may be a pig farmer and you may have a pig that sneezes on you, and that may mean that you're colonized with MRSA CC398, this livestock-associated form of methicillin-resistant Staph aureus. And that's bad for the farmer, but it may be that it doesn't actually transmit beyond that farmer. So we have this sporadic transmission of strains from animals or from the environment which don't go any further in the human population. Another type of risk that we can imagine, so we know we have these antibiotic-resistant clones circulating in our hospitals. Maybe they go out into the environment and back in again, so there's constant cycling. So this is in a sense a bigger sort of risk, that actually we have these big reservoirs of known resistant clones out in the environment. And a third, much more in a sense existential and difficult to quite quantify, is this idea that actually new resistant strains emerge in the environment. And of course, this does happen. We know it happens, but how often it happens and what we can do about it is another question. So just to illustrate these two types of risks, so on the left we have a pathogen or a bug or that's able to circulate within cows. It moves into the human population. It's equally able to spread within the human population. On the right, we have this kind of much more sporadic transmission. A case of one cow that's transmitted to a human, but it doesn't go any further, and the humans have their own strains which are circulating with the human, and there's not much intersection between the cows and the human strains actually. If you're thinking about plasmids, then of course it's a different thing, because the plasmids may themselves transmit independently of the strains. So it may be that we have sort of temporary intersection of microbiomes of cows and humans. And in that time, there may be time for plasmid, resistant plasmid, to transfer from the cow bacteria into the human bacteria. Or it may be that the cow bacteria can survive long enough that this transmission occurs. So we have to think about all these different levels of transmission simultaneously. And just to come back to this concept of multiple layers of transmission. So this is the Russian doll concept, which was first mooted by Shepard et al. Back in, when was that, 2016, from their hospital study, where they found a carbapenemase gene, KPC, which was integrated into a transposon, which itself was in another transposon-like element, which itself was in a plasmid, and the plasmid was in the strains. And all these things have different levels of mobility. So it's rather like this Russian doll. You have at the biggest level here, you have the actual bacteria itself. Within the bacteria, you have the plasmid. Within the plasmid, you have an integron. Within the integron, you have a gene. So what is it that we're supposed to be tracking? There are, of course, two distinct types of methodologies that we can use to sort of begin to address these questions. The first, which has already been mentioned, metagenomics. So I'm not really going to talk about metagenomics today, but I'll just mention a couple of papers later on, which I find particularly interesting. But of course, this is the method whereby you can look at all the DNA within a particular setting, if that's a river or if it's a microbiome, a pig gut or whatever, and look at all the resistance genes that are present within that particular setting. The other approach is just is a culture-based approach, where you take a sample from a particular setting and culture it out, and then you could look at actual individual genomes of individual strains. So this tells you a lot about individual strains. It doesn't tell you very much about the whole community. But the advantage, one of the advantages of this, is because if you've got the cultures, then you can actually do the phenotypic resistance testing as well. So another thing to bear in mind, that the presence of a resistance gene doesn't necessarily mean that those things are phenotypically resistant. So what about the plasmids? I haven't mentioned the plasmids in this slide at all. And there's very few, if any, well, there's one that I know of, a real proper, nice, in-depth epidemiological study on plasmids, and they're very difficult. I mean, they haven't really made it into the genomics era yet, because there's all sorts of technical challenges about plasmids. So you can't really, in a sense, so we can talk about replicon types and mob types and all the rest of it, but we can't. Up till very recently, we haven't been able to reliably and at scale assemble complete plasmid genome sequences from these sort of population samples. You can't do it from short read sequencing. So we're at the cusp of a new wave of data. I mean, being old, I've been through a few different waves of data from the MLST days to the short read genome days, and I think we're on another wave now of the long read data, where we actually can assemble complete, nice closed plasmid sequences, and this is really going to change how we view plasmid epidemiology. So we need the surveillance platforms to deal with those data, which we haven't actually got at the moment. But then there's all sorts of technical and conceptual challenges around typing. When can two plasmids be said to be the same thing? What sort of threshold levels of divergence are we interested in? Just the backbone genes of the plasmid, or how much do we take into account all the very variable accessory genomes? And we need, if we're thinking about transmission of plasmids in a One Health setting, then we need really intensive and deep sampling in order to really say anything very much, because transmission within these sort of landscapes is difficult, even when you think about strains. And, you know, big data genomics, it's all great, but there's an awful lot of biology involved here as well, which I think I've alluded to already, mentioning the ecology. But the fate of plasmids in natural environments is really, really complex question, and depends on all sorts of interactions between the plasmids and the bacterial hosts, and the plasmids and the wider environment, and the local adaptation of the strains themselves. So we have all sorts of different incompatibilities between different plasmids, incompatibilities incompatibilities between the plasmids and the host. We have addiction systems, ways by which plasmids can maintain themselves in a particular host. We have a lot of unknown questions about the fitness costs of plasmids, particularly resistance plasmids. What happens when you take the selection of antibiotic exposure away? Do the fitness costs outweigh become enough that the plasmids are going to be lost? We have all sorts of interesting community effects. There's a thing called the dilution effect, which refers to the fact that if you have a plasmid in a community of cells, then some of those cells might be able to acquire the plasmid, but actually aren't very efficient hosts for that plasmid. So actually they can act to sort of mitigate, to limit the spread of the plasmid. There are all sorts of environmental factors, temperature, pH, which can impact on plasmid spread. So this is the paper that I wanted to draw to your attention about plasmids, which was out when was it out? In March this year. And this is from Matlock et al. in Oxford. And they did a really nice, very in-depth population sampling around Oxfordshire in England of Enterobacteriaceae, and they compared strains from bloodstream infections, clinical isolates, all the plasmids within those isolates with all the plasmids from agricultural and wastewater sources. And they found they clustered their plasmids into 225 different plasmid groups, and they found that many of these were promiscuous across different bacterial strains. 4% were promiscuous across different bacteria. 8% of these plasmid groups were actually found within both human and non-human niches. So their conclusion was there's a lot of sharing of plasmids between the humans and clinical isolates and these agricultural and wastewater isolates. So let's just go back to two examples of papers that I just find particularly interesting, more or less plucked out of the air. They don't fit into the narrative of this talk very well. I just thought I'd mention them because they're kind of interesting. So these are the two metagenomics papers. This one was from the group of Frank Erstrop et al. It's a couple of years old now. So they looked at sewage samples on a global scale and looked at the resistance genes in those sewage samples from different countries and different continents. And they found that actually, so this is a principal component plot, and you can hopefully pick out the red squares, which are from Africa, the blue circles, which are Asian, and there's this open sort of diamond shape here, which is South America. So these ones all sort of cluster together a little bit. And if we look at the overall AMR load, it's highest in Africa and South America. So there's differences clearly between the low middle income countries and the high income countries. And most of this is down to the prevalence of these macrolizers, big brown bars here. So their conclusions actually was that it's basically down to socioeconomic factors and sanitation that is the biggest drivers of the differences in AMR on a global scale. So this is just one figure from this one paper. This paper is really, really data dense. And I've just plucked this one. Actually, it's like 2 panels from a multi-panel figure from lots of figures in this paper. But what they did is they compared the microbiome from pigs and humans, and HK and C stands for Hong Kong and China. So what they showed is, so in Hong Kong, almost all of the pigs that are eaten are imported from China. And what they showed by comparing microbiomes is that the microbiomes, in terms of the resistance genes, the microbiomes from humans in Hong Kong are more similar to the microbiomes from pigs in China than the microbiomes of humans in China. So because the people in Hong Kong are eating all the Chinese pigs, their microbiomes are more like the pig microbiomes in China than the human microbiomes in China, which I found really interesting. So we can see that when you have all this sharing, I mean, food's obviously a key route by which resistance genes enter the human population. Okay, so let's talk about this is more sort of what I do, which is these sort of more culture-based approaches, where you take a sample, you spread it on selective media, and you grow the colonies up and you sequence the genomes from it. So there's been a number of these whole genome studies on Klebsiella pneumoniae and E. coli over the last 2 or 3 years, that starting from just before COVID, really, that have attempted to sort of compare what's going on from clinical isolates with those from agricultural isolates in particular, but also those from other environmental sources. And in summary, basically, a lot of these have tended to show fairly limited overlap in the STs, in the strains, between human-associated E. coli and Klebsiella and animal-associated Klebsiella. But it's very difficult to be equivocal. These studies are difficult because you need to, first of all, you need to design your sampling frame really carefully. So there's all sorts of reasons why your samples from cows and your samples from humans might be different. If your clinical samples are just a sort of a convenience collection from the back of a freezer, and they may have come from diverse hospitals across the country or from different countries, may go back, have maybe decades, and all your cow samples are from a few farms near about that just happen to sample. So that's not a very clean comparison. So you need to compare like with like. So you need to compare clinical samples and environmental samples taken from the same place at the same time. And that's kind of a logistically difficult thing to do. And you need large sample sizes, because detecting transmission in One Health landscapes is much harder than detecting transmission in an intensive care unit. There's all sorts of reasons. You're never going to find all the transmission links. And there's all sorts of reasons why you may be misled into thinking, to having sort of false negatives, basically, missing transmission links. And it's, of course, as I've already mentioned, inferences aren't generalizable between different pathogens or between different geographical regions. Okay, so that's the first half of my talk, and now I'm just going to talk about a study that we published, which is very much in this vein, which we published came out last December, I think, in Nature Microbiology, large-scale genomic snapshot of Klebsiella species in northern Italy. So the headline here is that we also found this limited overlap between what's happening in the clinic and what's happening in the strains that are present in clinical isolates. Limited overlap between clinical isolates and animal-associated isolates for Klebsiella. Okay, so if you're not familiar with Klebsiella, it's kind of it's a gram-negative bug, kind of in the same ballpark as E. coli. It carries these beta-lactamases on plasmids that encode for carbapenem non-susceptibility. So KPC, NDM, VIM, and OXA48 are the kind of key classes of carbapenemase enzymes that munch up these carbapenem antibiotics, which are kind of the sort of last resort antibiotics, broadly speaking. So northern Italy is a hotspot for particular hospital-adapted clones which carry these plasmids, which contain these carbapenemase genes. The JPOMR Spark project, well, the aims were to look at the overall diversity of the Klebsiella genus. So this isn't just focusing on Klebsiella pneumoniae, this is focusing on all the other Klebsiella species as well, looking at this ecological question of how that diversity is partitioned according to the different settings, the different sources of the isolates. And then to look to see where those resistance genes are present in the environment, and then to finally try and infer something about the transmission dynamics of strains within and between the different settings. So these are where the samples were taken from. This is the town of Pavia in northern Italy. It's about an hour south of Milan. So this is the city here. So we took a kind of a broad geographical range around Pavia. This is on a larger scale map here. So all the samples were within a defined geographical region. They were all taken within a 16-month period by 3 incredibly hard-working people in David Cesera's group at the University of Pavia, who spent a year and a half doing all the sampling. So we ended up sequencing about 3,500 Klebsiella genomes. They weren't selected to be resistant. We just used selective media to pick out all the Klebsiellas. We didn't care whether there were Klebsiella pneumonia or other species. We wanted to look at, you know, if there's any transfer of resistance across the genus as a whole. So out of the 3,500 Klebsiella genomes, about half were Klebsiella pneumoniae, but in total we had 16 different species, and 3 of them were discovered actually during the course of the project. So it's amazing what you find when you start looking. So they're roughly equally distributed according to the human, animal, or the environment. So about 1,500 Klebsiellas from humans, from hospital, local hospital and community fecal samples, mostly collected from 5 hospitals in Pavia city. There's over 1,000 animal isolates, mostly from livestock, a lot from cows, but also poultry, pigs, and also quite a lot of wild animals. We took quite a lot of samples from flies as well, for example. And then the environment, we had lots from river water, from surface water, from plants and soil. So this is the tree, figure 1. So all the big blue part of that tree is Klebsiella pneumonia. This is just a mash tree because there's so many genome sequences. It take forever to do a Raxamel tree of all of it. This is just a simplified version of the tree. So this is a group that a new species that we discovered, which falls just outside the Klebsiella genus, which I got naming rights for actually in this one. So this is Superficibacter maynard-smithii. There's 2 other species here of Klebsiella, Pasteuri and Spallanzani. So they're named after French and Italian microbiologists. So I got to pick a British scientist for my species. This was isolated from the surface of an ATM machine. It doesn't have any sort of virulence or resistance properties, but it makes a very handy outgroup for the tree, so we kept it in. So that's the overall tree. In terms of resistance genes, we really didn't find anything outside of the hospital, outside of the clinical isolates in terms of the carbapenemase genes. So all the carbapenemase genes were within hospital isolates. And that goes for we phenotypically tested all the isolates as well. So none of the isolates from outside the hospitals were phenotypically resistant, despite this being a hot spot for these KPC genes in northern Italy. So they're all kind of either constrained in the hospital or they don't compete very well outside of the hospital. The sort of next level down, if you like, the ESBLs, CTXMs, and SHVs were present outside hospitals, but pretty much restricted to Klebsiella pneumoniae. Okay, so we didn't see a whole lot of resistance where we wouldn't expect to find it. This is a figure just showing all the different levels of resistance. These are all the different species. This is down here, abbreviations of all the different species. So you see most of them are low-level resistance. And then the ESBLs, we have this row here is the Klebsiella pneumoniae. So Klebsiella pneumonia with ESBLs are present in lots of different settings. If we look down vertically here, these are the carbapenemase producing strains are restricted to this part of the figure, which is the human hospital. These 2 columns here, human hospital. So the carbapenemase is not found outside and there's very few colistin resistance genes as well in these data. Where are the species found? Well, they're found all over the place, but also non-randomly distributed. So there is a lot of local adaptation in terms of the species, and this shouldn't be a massive surprise. So Klebsiella pneumoniae is associated with hospitals, also with humans, also with cows and pigs. So the red squares indicate an overrepresentation given a null of random distribution. There's a group of related species here, which are kind of seem to be associated with invertebrates, which was a new finding. And then there's another group here, which formerly called the Rautellas, which are associated with soil and plants. So that's not a massive surprise, except I should say pick out Klebsiella varicola, which is described as a plant-associated species, but actually we didn't find any on plants. And this is, again, another story about the type strain of Klebsiella varicola was found on a plant. So ever since then, it's been sort of considered to be a plant-associated species, but we didn't find any on plants. The different strains, this is now the same sort of figure, but on a intra-species level. So these are all the different strains of Klebsiella pneumoniae for all the different sources. And the same goes if we just consider the two most densely sampled settings, which are humans and cows. There are different strains. So the red boxes are those strains, STs, which are associated with humans, which are overrepresented with humans here. So ST307 and ST17, which are known human associated things. But we also have these cow associated strains. So there's difference. There's non-overlapping populations of strains in humans and cows. It's very clear. But there is transmission. So here's a case where all these pink nodes are from cow isolates from a particular farm, and embedded within this big pink clade here is one green node, which is from a human hospital carriage, so a fecal sample from a hospital inpatient. There's the hospital up there. Sorry, it's not the best map in the world. And there's the dairy farm. No, sorry, there's the hospital. There's the dairy farm. So we have no idea how that transmission occurred, but it certainly did occur. It's interesting. You actually do find clones in these dairy farms in these animal isolates. So there clearly is occasionally sporadic transmission going on. But rather than go around the whole tree picking out these bits of transmission, which would A, drive you mad, and B, would be a highly subjective way of doing it, we developed a very simple sort of pipeline whereby we first split all the data up into these groups, which are somewhat equivalent to sequence types, except not all the species have MLST schemes. So we used a program called PopPunk to define clusters, which are essentially like sequence types. We calibrated it to make it the same sort of size as a sequence type. And all the different colors here represent, all the dots represent isolates, and all the colors represent different sources. And we simply use different SNP thresholds to identify links within each of these clusters. So we use 6 different SNP thresholds between 0 SNPs and 20 SNPs. And then we just simply counted those links between same source and between different sources. And we did this non-redundantly. So we actually lost a lot of information, but there's a risk that of double counting if you don't do that. So we ended up with a matrix of all these different transmission events between different settings, and then built a network from that. So this gives you some. So this is an attempt to put weights on those arrows on that classic One Health flow diagram that I showed at the beginning, to actually try and figure out which transmission routes are more important than others. And we do see it's very clear that the most transmission occurs within settings, within farms, within hospitals. There's big black motorway here between humans, transmission within a hospital. And then you have another part of the network which is related to the soil and plants. So there are some constraints to transmission. And if we were to ask the question, where do humans get their Klebsiella from? Almost all the time it's from other humans. Okay, so that is at least in this setting. Okay, and that is kind of consistent with other work, similar work as well. So now just want to go on to the final the sort of work that I'm currently doing, and this moves on to slightly away from resistance. So I'll come back to the question of AMR, but it moves on to the question of virulence. And we should also, as we're doing these One Health surveys, there's no reason why we shouldn't think about virulence, presence of virulence genes in these different settings, as well as resistance genes. For Klebsiella pneumonia, the virulence genes have been pretty well characterized. So there's a small number which are very clear virulence markers. 3 of these, Yersinia-bactin, Aerobactin, and salmonellin, code for things called siderophores, which sequester iron. So iron is a key. The availability of iron is a key challenge for all pathogens once they get into the bloodstream. There's other virulence genes, RMPA and RMPA, which basically are involved in capsule. But these siderophores, or the genes encoding these siderophores, like the resistance genes, are associated with particular STs, particular strains. These siderophores are also associated with particular STs, the so-called hypervirulent strains. And rather than being in hospitals, these are community associated. So by and large, we have a set of strains that are circulating in the community, which are hypervirulent. We have a set of strains which are circulating in hospitals, which are resistant. And up till fairly recently, those two sets of strains have been kind of apart. So you're either virulent, a bug is either virulent or it's resistant. Now, the doomsday scenario that's unfolding quite rapidly is that these two traits are coming together, a process called convergence. So more and more Klebsiella pneumoniae strains are being reported that have both the virulence, these virulence genes, and the resistance genes. And more often than not, these are coming out of China, Southeast Asia, but not completely. There's a few cases turning up in Europe as well. So this is the sort of classical traditional view that we have the virulence genes here and the resistance genes here, but they are coming together. And they could be coming together through various sort of mechanisms, because all these, the virulence genes as the resistance genes, they're all born on plasmids. So it could be that strains are acquiring a strain which is already got a resistance plasmid, has acquired a virulence plasmid, or the other way around. Or it could be that the plasmids themselves are actually acquiring both virulence and resistant genes, right? Which would be the worst case scenario, because then those two traits will be able to be transmitted simultaneously. Okay. So back to the Italian data. So this is the same figure, that essentially the same format of figure I showed for resistance genes, but now for virulence genes. And I just want to point out this dot here. So this dot represents Klebsiella pneumoniae isolates from pigs, which all contain a virulence gene, eribactin. And this is what this was an observation which kind of launched off a whole new research direction for our group, in a sense. So eribactin is encoded by the IUC locus, which has 5 genes involved. It's on a plasmid. And there are a number of different variants of this locus have been described: IUC1, IUC2A, 2A, IUC3, IUC4, and 5. So the ones that are from the pigs is IUC3. They're all IUC3, so a slightly different variant of this locus. So most of the time, when you have a hypervariant Klebsiella pneumoniae infection, it's associated with IUC1 and IUC2. So there's not really any studies looking at the different functionalities between these two variants, whether they're equally as bad and equally as virulent, confer the same sort of virulence properties to the Klebsiella pneumoniae. So in all the the papers tend not naturally, many papers don't actually distinguish between these two variants. So they just say there's the presence of IUC or not in this particular plasmid or this particular strain. So in parallel to the Italian project, which I've been talking about, called SPARK, we were also involved in a One Health study in Thailand, which was driven by Matthew Avison in University of Bristol, which is next door to the University of Bath. And this followed pretty much the same sort of idea, except it was based in Thailand. And it was incorporated both Klebsiella and E. coli. So there were hospital patients, community carriage, strains from local farms, and also quite a lot of food isolates in this study, and soil and water. So there's food samples from 2 markets, all from this region, Ban Leng, which is a couple of hours or so away from Bangkok, in a northwesterly direction. So we found IUC3 in the Italian data, as I mentioned, in pig isolates. And actually, the association with pigs has been confirmed from different studies around the world, from different continents. There's a really super strong association with Klebsiella pneumoniae isolates from pigs with this particular IUC3 locus. We have no idea why. In the Thai data, we didn't actually have any samples from pigs because of African swine fever. We weren't allowed near pigs, but we did have a lot of samples from pork from these two markets. And just as in the Italian data, all the IUC3 isolates were focused on pig farms, in particular 3 particular pig farms. In the Thai data, we found IUC3 positive isolates from these samples from these two markets, which are only 2 kilometers apart from each other. And actually 13% of the pneumonia isolates from these markets carried IUC3, which is a really, really high percentage. A lot of those were from pork, but there are also some from poultry, in particular duck, and a few others. It's very difficult to equate that with the actual original source, of course, because they're on a market, so there's lots of cross-contamination going on. So we use long-read sequencing, nanopore sequencing, and we've now got about well over 1,000 complete hybrid assemblies representing both the Italian and the Thai data sets. So I'm just going to focus on a manageable amount of the data, which is the IUC3 plasmids. So we started off looking assembling 81 IUC3 plasmids because we wanted to look at the diversity, what else are on these plasmids, and they're roughly half and half from the Italian data and from the Thai data. The first thing to say is, so the replicon types, if any plasmidologists out there, so they're all INCEF1B and INCEF2, with a few other additional replicon types in some of the plasmids. So the Thai plasmids are bigger. That was the first observation that we made, quite considerably bigger. They also have a lot more resistance genes on them than the Italian plasmids. We then had a looked at where the resistance genes were on the plasmids, and we noticed that a lot of the Thai plasmids, not only they contained resistance genes, but they corresponded to this definition of convergence. So they are already virulence plasmids because they have IUC, but to be convergent, they also have to have an ESBL or a carbapenemase resistance gene. And this definition of convergence was originally put forward to describe strains, but it's equally applicable to individual plasmids. Okay, so something like 20% of all the IUC3 plasmids from these Thai markets also have ESBLs, so their definition of convergent, and these convergent plasmids are big. Okay, it's normally upwards of 200 kb, some are bigger than 300 kb, and the reason they're big is because they've acquired these resistance genes through a hybridization process with co-circulating resistance plasmids. So this process of co-integration is well known, that plasmids can basically fuse and come together and form one giant plasmid. So this is the first occurrence where we've actually been able to see this process of plasmids actually co-integrating virulence resistance plasmids actually coming together and hybridizing to form these so-called convergent plasmids. And we can see this because we've got all the hybrid data, right? So we've got hybrid genomes. So we're not just got all the virulence plasmids, we've got all the co-circulating resistant plasmids from those isolates from the marketplaces. And we found 7 really clean examples of this sort of plasmid hybridization process going on. So this is just one example. So we have a convergent plasmid here. Hopefully you can see the can't get my marker over. Oh, there we go. So this is the convergence. It's half blue, which is the AMR part, and half red. It's almost perfectly 50-50. And this is a plasmid which is 317 kilobases. It's a big one, from duck meat on one of these markets. The AMR plasmid, the blue part, comes from pork meat from the same market, and the virulence, the IUC plasmid, comes from an inpatient at the local hospital. And this is alignment, so there's a big inversion here, which kind of makes it look a bit more messy. But this is a big alignment. You can see it's basically the conversion plasmid in the middle. The red part here is the resistance. The blue part is the virulence gene, and it's basically just 2 plasmids that have come together as one. And we've got 7 examples, which are almost all as kind of clean as this one. And we look, yeah, sorry.
Can I just ask
We don't know. So they're predicted to be mobilizable. So they're or predicted to be conjugative, actually, not just mobilizable. So they have all the trigenes and all those bits and bobs. So mob type will actually tell you if they're predicted to be conjugative or not, and they all are. So there's no reason, we haven't done the experiments yet, but there's no reason from the bioinformatics that they shouldn't be. So we can actually map the presence of both the hybrid plasmids and the individual parental plasmids as to where they were found on the two markets. So almost all of we just use BLAST to identify the best hits for the putative parental plasmids in each of the convergent cases. And they're all, almost all of them are from these two markets, which, as I say, were only 2 kilometers apart from each other. So we've got this process of plasmid co-integration, convergence, which is known. There's a number of papers of, as I say, particularly from China, of these convergent strains. But because we've got all the plasmids, we can actually identify the donors. And what's more, we've shown that this happens a lot frequently outside of hospitals in One Health settings. Okay. Now, to answer your very good question, David, we don't know what the fitness consequences of this integration is. It may happen all the time, but these big plasmids may be lost all the time as well. We don't know what the sort of long-term consequences of these convergence events are, but we do find these links between the market and the hospital in terms of there's a couple of examples. So the example I just showed, actually, one of the parents came from a hospital. There's another example where the convergence plasmid itself is from a hospital inpatient, and the two plasmids were from the market. So there's that sort of more cryptic transmission link between the hospital and the market. Okay, so in the last couple of minutes, additional questions we can ask from all this data. So how diverse are these IUC3 plasmids in the context of all the other co-circulating plasmids? How are they distributed according to source, and human, animal, and environment? We can ask the same questions that we were asking about strains. We can now ask about plasmids, how they're distributed across ecology and how they're distributed across space. Do plasmids have biogeography? Okay, you won't find many papers discussing plasmid biogeography. So we can address these questions by, we went to the databases, we found all the other examples of complete plasmid sequences that contain IUC3, and we did a clustering procedure, which I'll show you in a moment. And we also identified, so these are 139 plasmid sequences, which include our 81, which are fully closed IUC3 plasmids. We then looked at all the short read data for isolates which have IUC3, and there's almost 500 of those. So we looked at those as well, but we don't have complete closed plasmid sequences in those cases. So this is a clustering procedure recently published by Sergio Alonso in the group of Jukka Karanda at the University of Oslo. So this is upwards of nearly 2,000 plasmids representing both the Italian and the Thai data sets. The green ones are all the IUC3 plasmids. So they represent a kind of a small fraction of the diversity of all the plasmids that are out there, and actually fit into there's 2 very clean clusters. If you can see, there's this cluster, and then there's a looser sort of cluster of clusters. So because we've got complete genome sequences for those, it's a fairly simple case to actually build a tree. We can build a plasmid tree from those IUC3 plasmids. And when we do that, the colors on this tree correspond to the clusters in the previous figure. So that cluster analysis, which is based on a sort of CAMDA approach, actually is pretty consistent with this tree, which is just, it's a SNP-based approach. So group 3 is one cluster, group 2 is the purpley ones right at the end here. This is another cluster, and this is this sort of looser cluster of clusters, which I've just lumped together as group 1. This was the big surprise to me, that if we then, so I was convinced, okay, maybe we can build plasmid trees, maybe that's looking okay. So we just took our biggest reference IUC3 plasmid, which is about 350 kb, and mapped all those short reads. Remember, there's about 500 or so isolates with just short reads which have IUC3, and just mapped all those short reads to that big plasmid, and we got essentially the same tree, those same 3 groups. And what's really striking is that one of these groups is very much associated. So the top red bar here is, so the top bar here, the red represent human isolates, clinical isolates, and the green represents animal isolates. And in the lower bar here, the red represents Asian origin. So this cluster is very much associated with Asian isolates and very much associated with clinical cases. So we're seeing subvariation in these IUC3 plasmids, which according to geography and according to source, and we've identified that this particular subvariant, which is very much associated with clinical cases, which raises the prospect that we could actually carry out some plasmids, because there are some plasmids in here which are European, so it's actually cropping up in European clinical cases as well. So this is a case whereby we can actually implement some proper plasmid surveillance to look out for this IUC3 plasmid subvariant. So finally, just to finish, coming back to this Matlock et al. Paper, they state in here that future 1L studies require dense and unselected sampling, near complete plasmid reconstruction. It's all about the long read sequencing or the hybrid assemblies, and to appropriately understand plasmid epidemiology across niches. So conclusions come back to the questions I asked at the beginning. There's lots of knowledge gaps in One Health. What are the constraints on AMR transmission? What is it that we're supposed to be tracking? So what seems to be true for one thing, strains in the Pavia study, maybe it's constraints on transmission that may not be true for the underlying plasmids. And how do we gauge the risk from AMR reservoirs, which I've just mentioned, haven't really explored. So in the Spark Italian study, we looked at transmission of Klebsiella in around a particular city in northern Italy. Although that region is a hotspot for this particular these resistance clones carrying the BLA KPC gene, we didn't find any of those carbapenem non-susceptible clones outside of the hospital environment. We found a lot of evidence for local adaptation and a lot of not so much evidence that there is transmission from one setting to another, which I would be tempted to put those two inferences together. So in the final part, so we found that this co-integration, this fusion of plasmids is very common in One Health settings, resulting in plasmids which are both virulent and resistant. We don't know what the fitness consequences are. We don't know so much about the mechanism. It's probably something to do with the presence of insertion sequences that encourage these sort of fusion events. And we found one particular subvariant which is associated with Asian isolates, and also with clinical isolates. And that's it. So just to thank the key players in my group who've done all the bioinformatics, Natasha, Marjorie, Samia, PhD students Samia and Kira, and lots of people on the Spark project. Particular mention David here, and now in Oxford, Angelica, who's a long-standing collaborator, and 1 million people on the ODART project, who are too many to mention, I'm afraid, but in particular, Matt Avison, who drove that project. Okay, thank you very much.
So thank you very much. I've been really interested. So I have been one of the graphics that you put this transmission between different human, for cats, dogs, etc. So thank you. And actually, there was this connection between humans and water. Oh, thank you.
Yes.
And that was turtles.
Yeah. And what, sorry?
Turtles.
Turtles. Turtles.
Because we had one case this in which one kid was infected with a multi-resistant bacteria, and we were sequencing around. We couldn't find anything, and finally it was the turtle. Turtle.
So there were two non-human sources which did actually have more evidence of transmission to humans than the others. One was water and one was dogs. So there's a lot of evidence that companion animals are a big reservoir of, we basically share the same strains as our dogs, which is frankly disgusting. So the turtles live in the river in Pavia, there's loads of them, and they basically shared the same bugs. We didn't see any difference between the turtles and the water bugs actually. Some of them, there was a big some of the turtle isolates were from a botanical gardens, so they all had a single clone which was swept through. So we kind of had to sort of disregard that because it messed up the sampling a little bit. But yeah, so I don't think it's anything to do with the turtle. I think it's to do with the water. Would be my.
Yeah, but before me to monopolize you, is there any question? Yeah.
Thank you for this very, very interesting, thorough talk. I wanted to ask regarding your protocol for the isolation and sequencing. Did you try to make a distinction between free living mobile genetic elements, viruses, and plasmids? Could you tell from your results if your plasmids were inside the cell or outside the cell?
So they will all be inside the cell because we're culturing.
Mm-hmm.
So we wouldn't have anything that is just free living we wouldn't sequence. So we're not doing any. So the studies that I've been involved with aren't metagenomic studies. So they're
okay.
single colony.
There is research that supports that we may be losing rhizostome information from mobile genetic elements they're outside the cell after cell lysis. So how do you think we could tackle that without having to actually perform mechanic filtering? Do you think there will be a possibility of doing that? I
don't know. It's a very difficult one. I mean, there's other problems that even if plasmids are within the cell, they can be lost very quickly on culturing as well. So I mean, it's a question of a known unknown, really. We don't know exactly what we're missing, and we just have to work with what we've got. I don't know technically how we would address that, to be honest, other than just to do the sort of non-cultural approach and just sequence everything that's there. I don't think you could do it very easily from cultural approach.
Okay. Thank you very much.
More question?
From Omar, there are two questions. So the first one, did the Andrews-Ken et al. Paper provide evidence for the second statement that improving sanitation would limit the global burden of antimicrobial resistance? It could be that the causation is reversed. For example, antimicrobial resistance and the high bacterial burden leads to poverty, which leads to poor sanitation rather than the reverse.
Yeah, sure. So there's an issue of causality there. No, absolutely. I think it was just the reason why I mentioned that paper was that I just wanted to give a nod to the fact that there's lots of economic and social factors that are important drivers, as well as all the genomics and all the biology that I'm talking about. But I take that point, yes.
And the second question is, what might a research program look like in order to determine which of the levels of complexity is the right unit of analysis for antimicrobial resistant transmissions? Or do you think that the answer varies by the context and the settings?
Oh my God. So I think that basically at the moment, we just have to try and work on all the levels that we can. And this is where the beauty of having long read data really helps, because all that data is there, right? So we can still look at strains. We can look at other aspects of the strains that we can't look at short read data, so rearrangements and so on. And then we can look at all the individual plasmids and all the integrants and so on and so forth. So at the moment, that's the best I can come up with, which is just to where possible, get hybrid assemblies. So hybrid assemblies are where you already have short read data, and then you can sort of choose the ones that you're interested in and generate long read data for those as well, and then mix the short read and long read to get nice. And we found this actually works really well with Klebsiella. I don't know how well it will work for other species particularly, but we found that we've got that working pretty quickly with Klebsiella. So I think so that's all I can say is just have a sort of whole evidence approach and try and just be cognizant of all these different transmission levels could be happening all at once. I'd have to think very carefully about designing a research program to actually prioritize which of those is going to be most important, because it's going to depend on the setting, it's going to depend on the bug, it's going to depend on the resistance genes themselves. So I don't think there is a single answer.
Any other question? I have a quick question. So when you mentioned these hospital patients, there were patients who were hospitalized because of an infection, or they were infected in the hospital, or
so
both?
half and half. So we, for the Italian study, we were kind of piggybacking on some routine surveillance that was going on anyway, which they had. So they were collecting the strains, so we sequenced the strains that we're collecting, and those were bona fide clinical cases, bloodstream infections to urine and so on. In addition to that, we were also taking fecal samples from hospital inpatients who could have been in for anything. So we distinguished between hospital disease and hospital carriage, and it was roughly half and half.